Yash?
No. In Canada, we actually have, in the eastern part of Canada, the local time is off by half an hour.
Oh, yeah, Newfoundland, yeah.
So it's crazy system.
Yeah.
Oh.
Well, Yash.
Whoa.
Wait a couple minutes for everyone to get here. It might be just the three of us. Kind of kills what I was gonna. Well, I can talk about it anyways. But people watch a recording of it. It'll be easier to.
I'm gonna get a drink.
All right. It'll be easier to kind of revisit it. Hello, Ujjwal.
Hello, Alicea.
Hi.
Hi.
Okay, well, why don't we get started, and then people show up later, we'll let them know what's been going on. So, welcome to this week's meeting. I would ask if anyone had any news or events they'd like to announce. No, nothing. Okay. Let's see. Today we don't really have a speaker. I have a couple of papers. I'll mention that I found. Kind of like to go over them, and then we have we want to revisit our task board, our GitHub issues board, to see if there's stuff that we can put on or update accordingly. So we should do that on a regular basis to make sure we're coming along on different things. So the first thing I'll do is the papers repository here. So I have a new set of papers that I found. I always have like this reading queue. Let's see. Dick says a possible interest. Yes. So Dick's been working on the diatom bibliography, and so this is in the chat.
Okay, just a second. I have a phone call. Just a second.
Okay. So Dick has a diatom bibliography he's been working on, and it just has a lot of references for diatoms, different readings and different references, academic references, and so forth. And so it's downloadable from this link, And if you have EndNote, you can open it up and look at it. There are other, if you don't have EndNote, I think you can get a copy of EndNote, like a educational version for free. I don't know if you can open the files in some other bibliography manager, but if you want to take a look, you should go to this link. Actually, why don't we visit the link really quickly to see what's at the link. So this is his Sync. It's a platform called Sync, so it's like Dropbox, and so you might have to create an account to get access to it. But we have the instructions and then the folder here, which has the references in the bibliography. So that's of interest. There's Jesse.
Major point is keep you from touching your face.
So that might be of interest to people.
Okay, see you.
Hello, Jesse. How are you?
For some reason, detective wants some blood work, which they wants immediately.
So, all right. So why don't we get into the papers? I had mentioned this.
For 4 hours before, but it's early enough.
So let me share my screen. That's what I was forgetting to do here.
She said about you wearing a mask.
So no problem with other people coming.
So people will see my screen here. Can you see this repository? I'll put a link to it in the chat. So put that in there, and it should be shared. If it isn't shared properly, just send me a request for access. Jesse says, "Morning, everyone. Staying healthy, or at least trying to." Well, good luck, Jesse. Everyone is trying to stay healthy. So these are the 3 papers that I found. These are actually, well, I'll start with the first one. So 2 of them have to do with neuroscience, and the other one is about modeling. And I thought Jesse would enjoy the 3rd one, especially. This one is Philosophical Transactions Be Hierarchy in the Organization of Brain Networks by Klaus Hilgetag and Alexandra Goulas. Alexandros Goulas actually was, so in my other group, I have a student who was working on his Brandenburg Vehicles platform, and he visited the INCF conference in Warsaw last year, and he met this guy, Alexandros Goulas, who was very interested in the work that we were doing. So I never really got in touch with him too much, but he does this sort of connectomics type research. So it'll be, okay, 45 cases of COVID-19 in India. Sorry about that. So let's go back to this. So this is the paper Hierarchy in the Organization of Brain Networks. And so they talk about how in systems neuroscience there's this concept of hierarchy. And so the idea of hierarchy is that there are levels of different things, and they're arranged in a way that sort of suggests function. So your top level is the thing that maybe controls lower levels. There are a lot of different architectures that one can use for hierarchy, but the idea is that there are top levels that are above, lower levels that are organizationally different. They have maybe they have their scales, their spatial scales, or their functional scales. And so that's what hierarchy means. So, however, this concept has been interpreted in many different ways, which are not well aligned. Which is for sure. We talk about hierarchy a lot, but we don't really know what it means in a specific context, and what if there's their general principles of hierarchy. So this observation suggests that the concept is ill-defined. Using the example of the organization of primate visual cortical systems, we explore several contexts in which hierarchy is currently used in the description of brain networks. And so they come up with at least 4 different uses. So the 4 different uses are as 1, a topological sequence of projections. 2 is a gradient of features. 3 is a progression of scales. And 4 is a sorting of laminar projection patterns. So, I mean, not getting into what those exactly mean, you can think of it in 2 different ways. One, we have something, they're saying something about connectivity. So they're saying something about like how projections are made from one brain area to another, and that it's organized in an orderly fashion. And 2 is that there's some sort of like spatial scalar thing going on, where you have smaller areas feeding into larger areas, or some sort of like, some sort of functional hierarchy, where you have one area that feeds into another area that does something like of a higher level. Like in visual cortex, it might be processing, features like blobs or stripes, and that feeds into an area that processes shapes. And so, if you know anything about neural networks, you know that in deep learning or in some neural networks, this is precisely how features are analyzed. So you get like smaller, you get like, basically more fundamental features like stripes or blobs, and then it feeds into a processing of shapes, and then maybe a processing of the entire image. And so this has a functional consequence of kind of, breaking up how things are processed. You'd say, well, why don't images, say, get processed in one shot? And the answer, of course, is that, well, there are a lot of different types of features that maybe need to be accounted for before the entire whole can be assembled. And so that's why you have these hierarchies. So this paper, though, clarifies, I think, at least some of this, and maybe grounds us in terms of when we use a term like hierarchy, what do we mean? And, gives you a clearer picture of that. So hierarchy is one of the most popular terms in current network and systems neuroscience. In a PubMed search of the keyword hierarchy, along with brain and network, so they searched those 3 terms together, yields more than 2,500 references, with a strongly increasing frequency over the last 15 years. And so this has become like the idea of hierarchies in brain networks has become very popular. And 2,500 references is a lot for a very small area of the literature, but it's been only over the last 15 years. So, and again, people have been talking about this for years, but they haven't really, it's been very vague in terms of what people are thinking. So this is an example here. This network, this is what they call a visual cortical hierarchy. And this is based on an image from about maybe 30 years ago, maybe 25 years ago, where they were doing studies, in macaques, which are a type of primate, where they would map all these connections out by hand and then assemble these networks. So I don't know if this is actually based on there might be some neuroimaging involved in this particular image, but a lot of the early neuroscience, they didn't have neuroimaging, and they didn't have like whole brain neuroimaging until fairly recently. But even with like, studies where you like put in different types of blockers and do different types of behavioral experiments, they've still been able to come up with this type of map. And so this is a pretty complex map. It just gives you an idea of how complex our visual system is, because this is very similar to the human visual system. And this is how people are thinking about it. And there are hierarchies within this network. So if you look at it closely, you can see some of the hubs of the network, which are just areas that are more connected than other nodes. Then they talk about the topological arrangement of different areas of cortex. So if you're interested in cortex and how the different areas are defined and connected, this might be an area to look at in this paper. talk about the plan of the present paper. Okay. So they actually do a little bit of experimentation here, where they look at the sequential topology of the primate visual system versus benchmark networks. And so they just look at the connectivity patterns within primate visual system versus some other types of networks. And this is an interesting set of experiments that people will do to look at to assess the structure of a network. So this is the primate visual system, and this is something they call a circular network. So the nodes are arranged in a circle, and then you plot the connections between the different nodes in a way that they're described in the literature, and you get this pattern within the circle. So you see these lines going across. So, I mean, it looks pretty complex, but there's no way to assess what that means, because it's like, okay, there's a connectivity pattern. There's some darker areas where the lines overlap a lot. There's some lighter areas where they cross here. Well, what does that mean? And so you can actually assess a network like this versus a number of different types of connectivity. So in this experiment, they're using something called the most sequential network, which is where all the neighboring nodes in this network are connected. But there isn't I don't think there's any long-range connectivity. So if you can see the connections here along the edge, there are all these lines along the edge, but there's nothing crossing the circle. And so this might be where you order your nodes by some criterion, and every neighboring node in this arrangement is connected, but no one else is connected. And so they're all connected, but indirectly. The primate visual system doesn't resemble that one. But what about a randomized network? So that's where you have these nodes that are like arranged in a circle, and you just throw random connections between them. And so you say, well, how many connections does the primate visual system have? Well, we'll generate that same number of connections, but it will all be at random. And so again, this primate visual system doesn't resemble the random result either, because the random result seems to be more, if you look at the lines going across, it isn't as irregular. I mean, there's sort of an irregularity, but there seems to be a lot of crossing across the circle, and it doesn't look like this. So then you go to the most distributed. So then this is not randomized, and this is not most sequential, but this is, I guess, where nodes are connected to things that are the farthest away from it. And so it's most distributed. So you try to break up all the local connections, and you have this most distributed. And it actually looks, maybe a little bit more pattern than the randomized one, but it resembles a randomized result. But still, the primate visual system doesn't resemble that either. So you can do experiments like that too, and then you can also assess the sequential cost, which is the cost of a connection from point A to point B. And you can see the primate visual system is in between the most sequential and the randomized, and has a little bit lower sequential cost than the randomized. So those are ways you can assess networks like this. And I don't know, maybe people have heard of like the idea of small world networks and scale-free networks. You can do the same thing with small world and scale-free network connectivity. You can create a plot of like maybe an idealized small world or an idealized scale-free network, and compare it to something like the primate visual system. And you'd find that there'd be differences there. You can assess it visually, or you can assess it with some metric. It could be cost, path length cost. It could be like with something they call centrality, or between the centrality, or some other measure of connectivity. If you're not familiar with complex networks, there are a number of different measures that people use. They put a number on the network in terms of like its relative value relative to networks that are randomized or scale-free or whatever. And so I thought that was interesting, because I don't know if anyone's in this group has seen how they assess network connectivities. But that's basically the method. And it's interesting because a lot of biological networks are kind of unique. They don't really fit into any of the theoretical models. I've been kicking around this idea with another collaborator about writing a paper on that, actually, where you have like these network topologies that in biological systems that are unique to a lot of the things that we look at in network theory, like, the different types of networks I described to you, that they don't really resemble any of those. And so the question is, do they really fit into that sort of typology, or do they have their own set of rules, perhaps, or they are just, unique in terms of their connectivity? And so if you're interested in this, we can have a discussion about it, offline or in subsequent meetings. In fact, I might revisit that at some point. But so then there are some nice figures in this paper, like something they call architectonic gradients, which are just kind of like maps of like, how things are connected in different layers of cortex. So I think Jesse was talking about he was interested in the layers of cortex. And so this is maybe something to look at, again, in macaque cortex. And then they revisit this idea of nested modules in the brain. So you have connectivity in different parts of the brain, but those may be, different regions, but there are neurons within those regions. And so you can look at like connectivity between neurons, connectivity between regions, and then, get an idea of the different levels of connectivity. So this paper, I'm not going to go any more into it. There are some really nice figures, and it's a really good paper for understanding this whole idea of hierarchy in cortex. Got a message in the chat, and that is yes. Okay, Jesse said yes. So that's that paper. Then there's this paper, Cortical Layer Formation in Development. So the title is Creative Destruction, the Basic Computational Model of Cortical Layer Formation. And one of the authors is Marcus Kaiser, who works on something called developmental connectomes. And this is where he's looking at how connectomes or brain networks develop in development. And he's written a number of papers on this topic, most of which have been theoretical. I've been keeping track of his work. It's really interesting. He has a lot of interesting ideas, but this is one of the papers one of his newest papers that he's put out. So this one is, the abstract says, one of the most characteristic properties of many vertebrate neural systems is the layered organization of different cell types. So it relates back to the paper we talked about. This cytoarchitecture exists in the cortex, the retina, the hippocampus, and many other parts of the central nervous system. And he's talking about in humans or in mammals, where you have different layered. So in cortex, it's a 6-layered structure. In some other parts of the brain, it's a 2-layered structure. And so those layers are, they're interconnected and they have different functional roles. And so here we provide a general computational model for cortical layer formation in 3D physical space. We show that this multi-scale agent-based model, comprising 2 distinct stages of apoptosis, can account for this wide range of neuronal numbers encountered in different cortical areas and species. So there's this idea that brain size in different mammals is controlled by the growth of this cortical sheet in terms of size. And one of the ways that happens in development is that you get, cell proliferation, but also apoptosis, which can sort of be like a counter to proliferation. And so apoptosis is something that maybe you don't know the term apoptosis. It's a programmed cell death. So the cells will be born and they'll divide and they'll remain alive for a while, and then at some point they may die in a programmed way. So they might divide a certain number of times and then die off. Or they might die in neurons. Sometimes they die if they're not connected into a network. If they can't find a projection to another neuron, they die off. And so this actually has an important role in development. in terms of shaping where cells, are in the body, but also the number of cells. And so that's something that they talk about here. And so they use gene type rules. They use different, they use an agent-based model for cells, and then they use gene type rules to sort of as a sort of a control for like apoptosis and things like that. So let's see this paper go through. They talk about the cortical nerve architecture. They talk about they have a number of so the materials and methods. What they're doing here is they're doing some experimental verification of neuron numbers. So they look at some histology, and then they have a simulation environment. So the simulation environment is the open source framework CX3DP. I don't know what that is, CX3D. It's some sort of open source software that they're using for this. They implement an agent-based model using a parallelized version of this program. So they actually use genetic regulatory networks as a base. So each cell will have a genetic regulatory network, which is a series of genes that are made up, and then they have an expression of something. So like in a GRN, you'll have a number of different genes, which could be like a string of binary values or some other switch, like a set of states that you can switch to, and then it behaves like a gene regulatory network. So you'll have like a stimulus that might stimulate the expression of a certain gene. Genes will express other genes, and then there's some output that relates to the phenotype. And so it allows you to regulate things in the cell using this program. So you can define a cell state by using one of these GRNs. They can be simple or complex, and they give you they try to simulate the genome. And I mean, it's not like a very complicated model of the genome. It's a very sort of a very low level abstraction of the genome, I guess. You just have maybe a couple genes, and you have some rules that determine how they're expressed, but it does sort of simulate how the genome expresses itself. And so they use this type of model. They do the simulations. Then they also get experimental data. Well, this is from the histology. So this is where they determine layer thickness. So basically what they do is they're parameterizing this model using data. They're using observations from row brains. And then they use the simulation to simulate things that don't necessarily exist. So this is all in the methods. Then the results. So let's look at some of the figures. So they have a number of different, let's see, schematic of sequence of cortical layer formation. During cortical development, progenitor cells proliferate and differentiate into different neuron types in a consistent sequence. So this is what they're finding here. So they're actually looking at different states of the neurons and how they progress using this genetic regulatory network. So they have this is a state diagram, and they're looking at different states. So arrows indicate probabilistic paths on the network consisting of the state diagram. The initial purely proliferative state S1 leads to state S2, where an internal timer triggers the state transition. From the second proliferative state S2, neuronal migration of MZ cells is triggered with probability CM. The marginal zone is the predecessor of layer 1. And so they have this, they come up with this map of different states of how what neurons go through in development. They do this, they derive this, and then they apply this to the simulation. Then let's see if I can get some real good results here. Okay, so I think this is the number of neurons under 1 millimeter squared of cortical surface area in the cortical layers of different areas and species. The shown examples are human temporal cortex, rat somatosensory cortex, mouse barrel cortex. So there are a bunch of different species in different areas of the cortex. And so they say that there's substantial variation in the dimensions of individual layers and the relative proportions within the same cortical area and species. This variation in cortical layer thickness stems from differences in neuronal size as well as cell numbers. Let's see what else here. And they have the spatial distributions of neuronal locations, layer-specific neuron numbers in different species in different areas. Then this is, I think, the simulation neuron numbers in individual layers during layer formation. So they kind of go through this, they kind of characterize the data, then they do the simulation. Let me see, discussion. So the presence of an abundant variety of layered structures is a common property of vertebrate brains. Although a fully comprehensive picture of cortical structure and function is currently lacking, developments in neuroimaging techniques have enabled key insights into these relationships. And that would be mainly between laminar organization and electrical activity. So a mechanistic, in-depth understanding of how cortical layers arise is currently lacking. So we can use computational models to inform this, but we don't really have an answer, I guess. Conceptual layer work is different from traditional models in that it follows an agent-based approach. Our model does not rely on any pre-specified neuronal architecture and is initialized solely from a homogeneous progenitor cell pool and a single extracellular substance gradient. So they're generating a phenotype in a self-organized way. So this. I mean, if you want to learn more about this paper, you can go read it. I think it's a pretty interesting paper, and I'm probably not doing it justice by kind of walking through it this way, but I just wanted to give you a taste of it. So we have another comment in the chat. Okay, I have a figure to show when you're done with this paper. Okay, Dick, do you want to give that figure?
Okay. Let's see. We're sharing on this thing.
In the lower. Oh, I can stop sharing actually. Okay, go ahead.
Is that? No, there it is. Okay. There it is.
All right.
Can you see this graph?
I can't see the screen yet.
Can't see it yet.
If you shared it.
Can you see it now or no?
No.
No.
If you go down to the lower right-hand corner.
Yeah, it's present now is grayed out.
Oh, well, I don't know why. I'm not presenting anything. Huh.
Settings is one for. You're stuck. Okay. Okay, why don't you go on? I will prepare a PDF and send it to you by email.
Okay.
You can show.
What was it about, by the way?
Oh, it's size of brains versus geological time.
Oh, yeah, that would be interesting.
Okay.
Yeah.
Oh, I see what I did. I didn't. There's a window I didn't see. Okay. Select window. Oh God. Oh, it gives me 50 options.
Oh.
That's no good. Oh, there we go at the bottom. Screen 2. No, that's not it. Okay, I'll try again.
Okay.
You're supposed to know the numbers of your screens.
Yeah.
Your entire screen. Okay. I must grant permissions in order to screen share. What is this? I'm hitting present now. Present your entire screen. And I'm getting, you must grant permissions in order to screen share.
Huh.
It doesn't give me anything to do except dismiss.
Oh. Well.
Communications. Let's go back to telephones. Rotary dialogues.
Yeah. Dick made a comment. The number of cell types in the brain may exceed the number of cell types in the body. Yeah, we've had some discussions about cell type number in the past, especially with respect to like the human body. And so, I mean, when I talk about cell types, they're, you know, that's an interesting point, is that we don't really know all the cell types, or, you know, it's sort of a decision we make based on what we know now and what we can use as a criterion for understanding what the cell type is. So, you know, people have used, microscopy images. They've looked at them and they've made determinations about cell type. Some people have used molecular methods. And when you use molecular methods, it's actually not necessarily more clear. It's, it's a different criterion, and you have to still make the same decisions about what constitutes a cell type, what are its properties. And so it's a very hard problem no one's solved. So when you see a simulation like that, you see that people have made decisions about what the cell types are and what what defines that cell type. So I'm going to go on to the last paper, and you can send me that in a PDF and I can distribute it to the rest of the group. But this one I think Jesse might be interested in here. Actually, let me share my screen again. So this paper is about modeling, and it's called "Modeling is the Practice of Representation." And when Paul Fishwick was at the University of Florida, I took his class on modeling. So this was like years and years ago. But he's an interesting guy, and he writes a lot about simulations and modeling. He's a computer scientist. So they talk about one of the characteristics of being human is the model. So his approach to modeling goes way back to like cave art and other types of sculptures. So like models are at their base, they're ways to represent things in nature or in the world for humans. And that can go back to like cave art paintings where people were painting, like different animals and different scenes, or it could be like sculptures where people create sculptures of other people or animals. And the whole idea of modeling is to, make a representation of that. Okay, I'll check my email. And so modeling is ubiquitous. It's not just something you do in a computer. And so he's making this claim that models are natural transformers from human experience to information. To create information for object X, create a model of X. Even simplistic activities such as grabbing a drink from the refrigerator or taking a walk translate into information management and processing. And so people sometimes, for a lot of, we don't talk about human-computer interaction in this group, but a lot of human-computer interaction is precisely that. It's like modeling an activity like, using a computer, or in terms of the Internet of Things, it might be like, having a wearable device and walking around the room, having a representation of that room. So it defines a model, and that's always a fun exercise, but you have, kind of goes through that. I think he does a pretty good job of that. The idea of representation is common in these definitions, and we will suggest lies at the heart of defining model. So he talks about it in terms of representation. He talks about modeling with numerous colleagues, since in most disciplines modeling is already present and activities by differences in vocabulary. So it's sort of a way to clarify like a representation of something. Linguistically, you could say something that's very difficult to understand, from one person to another, but models might be able to consolidate that meaning a bit. So they've taken a hybrid cultural, historical, and multidisciplinary approach in this paper. And so I think Jesse would be really interested in reading this all the way through. It's just like just a position paper on modeling and the worldview of modeling. And I think it's really interesting. He puts a couple of points out there. Modeling is a cultural phenomenon. Mathematics is a mental framework for abstraction. Computer programs are models of intelligence. Modeling transforms reality into information. And then they make a couple of claims here. I'm not going to go into the claims, but basically they stem from a lot of those points that he mentioned. I think that's a pretty nice paper. If you're interested in modeling and sort of maybe the philosophy of modeling or other types of things. And then, of course, he has this new class, Creative Automata, which is, I know he was doing something called aesthetic computing years ago, which is where you you use art to represent different systems. So you can represent different systems using like an artistic representation, like you might use a sculpture to represent like having dinner, or you might use like a stereo, with moving parts to represent some sort of computational process. Those sorts of things. I think that's the idea. So that's really interesting stuff. I would look into it a little bit more. I don't want to check my email right now. We'll do it. I'll send it out to everyone after the meeting, and we'll probably talk about it next week. So why don't we move on to the major tasks for 2020? I know I spent a lot of time on those papers, but it's okay, I guess. So let's revisit our tasks. So we have a couple things in the hold and to do and progress categories. Some things that are finished. So today I'm gonna record George's presentation on structuredness and entropy. I invited him to give a talk, and next week hopefully we'll have the talk and I'll play it in the group, and we can see he published a paper recently on this topic, so he's going to present on this. Jesse presented on his natural selection finds natural gradients, did a blog post for Open Data Day, which may need to be transported into this board, some of the points from that. I haven't done that yet. I'll probably do that after the meeting. So in progress, we have the CompuCell 3D model for C. elegans development. And I think that was something that Ujjwal said he was interested in. Okay, I present on so we'll present on that in the coming weeks. So why don't I make a note here? Okay, we can put that into do. We can create a convert to issue. And so we used to link these in one of our summer sessions last year. We used to link issues, but I'm not going to do that right now. But these two things are linked. So that's good. Schedule presentations. So I've been doing this. That'll continue as we get people interested in presenting. Anyone's welcome to present. Just let me know. We can put it on the schedule. The axolotl montaging. So I actually received on a zip or a USB drive here data from Susan. from from Canada, and she sent this on a disc, and I've been looking it over, but I haven't really gotten into it that much. So this is something we'll have to revisit. Let me put it on this as so it's on a USB drive, and it looks good, but we're going to have to talk more about how to what we can do with it. Actually, I should say axolotl data, which is maybe more accurate. Let's see. And so we'll do that in a coming week. And then the bacillary and non-neuronal cognition paper. So we talked about that last week. I actually spent some time putting together an outline on that. I don't have it ready to share yet. Actually, maybe I do. Let me see if I can find it in the drive here. Okay, this one. I gave it like a little working title. I don't think that's going to be the title, though. Bacillary psychophysics. But this is the outline that I've come up with. Kind of just stream of consciousness. But we'll talk about this in future meetings here. What we want to do. I think the models of movement that we talked about and that Dick presented in an earlier meeting, maybe that's worth revisiting, but in terms of like the plausibility of the different models, and then applying maybe, or talking maybe about neuronal regulation, different types of neuronal regulation that might explain these models of movement. So you have a bunch of models, you say which one's the most plausible, then you look at those models and you say, can we think about it in terms of like a model of neuronal regulation, which is, there are things like Hebbian learning or entrainment that brains do that we can apply to this. And then there's a hypothesis, and we distinguish between real neuronal behavior that's produced by a brain and neuronal behavior defined by these points up here, not produced by a brain. And so there are a couple people that have like kind of proposed things along this front. At the OpenWorm meeting a couple years ago, I think it was Matteo Cantarelli, he posted this image where he took like a video of C. elegans moving around in a dish, and then he had a simulation produced by his Geppetto simulation. And he said, can you tell them apart? And so he proposed that you develop a Turing test for distinguishing these two types of behavior. So in other words, the simulated behavior, how do we know it's real? Bless you. And there's, potentially a Turing test there that can be used to distinguish between, real life behavior and simulated behavior. And then the same may hold true, say, for like something like a bacillary, which doesn't have a brain per se, and something that might have a brain that would produce the same behavior. But there's also another thing that people have talked about, which is the last A-Life conference had a fake life recognition contest. And that's where they actually took artificial life representations, and they wanted, they gave them to people and asked them if it was real behavior or not. And so they were able to kind of, they wanted to see if it was lifelike or not lifelike, and they wanted people to assess it and all this. So there's definitely something going on at that interface between, simulated life and fake life, and, if it's produced by a brain or not by a brain. So, I mean, that's what I'm thinking in terms of this. I think I have this shared. I don't know. I don't think, maybe I don't have it shared. Let me adjust the sharing on this, and then I will share with you the link. So, edit, yeah, okay. the sharing should work. And if you can't get access to this, yeah, just request access. So this is for this outline for the bacillary behavior psychophysics paper. So, I mean, you can comment on it or add sections or suggest changes. So it's up there now. So that's in progress. And we'll talk about that in coming weeks. This is something that stems from this blog post in Open Data Days. So one of the things I put on here is reorganizing DevoZoo. And it's something I mentioned, I think, last week. That's on hold. I just put it on hold because I don't know the path forward on that. If people are interested in this task, we should discuss it further. But it's ready to go into progress. And then our to-do, we have review axolotl data. We have that already. Present on 3.3 CC3D progress. This idea of computed tomography via cloud computing, this is something Dick brought up a couple weeks ago, and no one took him up on it, but I'm bringing it up so that people might remember and maybe be interested in it again. We have Dick. If you want to do that, you can talk to Dick, and then we can talk about it in the meeting. Embryo visualization for Docker. I think we had mentioned that. I think Jesse and Ujjwal were interested in it at least. That's something again that requires more thought. If you want to, if you have more thoughts on it, please share them with me. Someone's coming in. Okay, I think Dick got knocked offline. So then the complexity measures, which is something that I think we talked about briefly, and I don't remember where we stood on that, but that's an open issue. And then I'm thinking of giving a lecture on the differences between PCA, UMAP, and TSNE. And these are methods for multidimensional representation. And I know those may be just like a word salad right now, but we do a lot of stuff with multidimensional data in the group. So I was thinking of giving a short lecture on the differences between these. Because if you read a paper now, like if you read a molecular paper, they'll often use either UMAP or TSNE to represent the high dimensional data. They'll take a bunch of variables and they'll boil it down to a couple of variables, and they'll create these really nice looking plots. They're just like these beautiful plots, but they don't know. No one really knows what they mean. It's kind of fascinating. It's like they're like these things that have a lot of. I think I was talking about this in our Saturday meeting. They have a lot of beauty to them, and they're very intuitive, but nobody really knows what they mean. And so in one of the coming weeks, I'm going to give a talk on this. I also wanted to put another thing on the to-do list, which was, so I think this is something that Dick asked me about, like at the beginning of last year, and I didn't do it. I just remember this because of all the virtual conference talk now. But Dick used to run a course called Embryo Physics, and it used to take place in Second Life. And there's a lot of interesting stuff there. And one of the things we might do is archive a lot of those materials to some other format. So right now there's like, the stubs that have the description of the talk and like an abstract. But I think he said that he wanted to archive this in a better place or revisit some of the lectures. So I'm open to that, Dick. If you're still interested in that, we can plan out a strategy of how to put that somewhere where it's accessible, maybe work on it a little bit to get it organized in a way that's more accessible to people. Because I remember that course. That was a very good set of courses or set of lectures that resulted from that course. Our old webpage is still online. I'm not sure. But we might actually be able to get it either from those or from the Wayback Machine, which is it's one of those things where you can find web pages that were live like maybe 10 or 20 years ago, and they usually have all the details still there. But yeah, we can look through it and see. I was wondering if, Dick, do you still have access to that Second Life area that we used to use for the embryo physics course? Okay, put that as a note. All right. Okay. Oh yes, but I'm rusty. Yeah, I know. It's like I usually don't spend. I haven't been in Second Life in like 5 or 6 years, and yeah, you have to. For those who've never been in Second Life, you have to like navigate your avatar through a virtual world, and sometimes you can fly from place to place, and you move around. Sometimes it's like really glitchy, and you end up bumping into things. And but yeah, well, I mean, we'll see where those materials are. We'll maybe try to get them together for that. But so that's the list right now. I'm going to take off of this blog post, and I don't know if people have seen this in this group. I think I mentioned it to my other group. Now there's a blog post for Open Data Day, and I laid out some of the things to be done. It's just a rough list, and those things are going to be put on the major tasks. We already have the DevoZoo. This is a link to the blog post. We already have a DevoZoo thing. The DevoZoo maintenance is mentioned here, but there are other things as well. And so we'll see how far that goes. Like I said, Open Data Day started Saturday, but it lasts infinitely into the future. Probably revisit things next year on Open Data Day to see where we are. So, is there anything else anyone else wants to mention before we go? We're at the top of the hour. Okay. All right. Well, good. That's a very good meeting. Next week we're going to probably hear from. Okay. Yeah. That's great, Jesse. We'll hear from George Mikhailovsky, who wrote a paper on structuredness and entropy recently, and he published it in, I think, Entropy Journal. But he's going to give a talk on that paper. So it's a very thick paper. It's a very long paper, has a lot of figures, and it might be a little bit inscrutable for people. Wanted on Bacillary psychophysics. So yeah, if you're interested in that topic, I put the link to the outline in the chat. And so if you're interested in participating, just join into that document and please make comments on the side before you move text around, just because it gets really messy inside. But if you want to put in text, your own block of text, you may. Okay, Jesse, yeah, check it out, please. So we'll hear from George.